Hello everyone, I am Akriti and today we are going to study about cell. So first of all, the cell chapter in AP biology is very important as it contains most of the questions of the free response questions as well as the MCQ part. So the cell unit may include 20 to 25 marker questions. So first of all, what is cell and how the structure of the cell looks like? So here you can see there are two types of cell. One is the typical animal cell and the other one is plant cell. So as we know that all the organisms are made up of cell. So organism, they are made from the organ system. Organ system is made from the organs. Organs have been made from the tissues. And tissues are made from the cell. So cells are known as the building block of life or the functional and the fundamental unit of life. So when we are going to study about the cell, three questions should be clear in your mind. So let's try some questions. First is who discovered cell and how? You should know this. I hope you know this answer that Robert Hooke discovered cell in 1665 when he was observing thin slice of cork. So the cell which Robert Hooke discovered is dead cell. So sometimes question comes to confuse you that who discovered the living cell? So it was Van Leeuwenhoek. He observed pond water and he was the one who discovered living cell. Second question, which is very important and is a basic of the cell chapter, is the cell theory. So cell theory is given by Skeldon, Kohn and Virchow. Cell theory states all living organisms are made from the cell. Second, cells are the basic unit of all organisms. Third, all the cells arise from the pre-existing cell. And the third question, which is very important, is why cell is known as a structural and functional unit of life. Cell is the smallest unit of life. Cell is required to produce the organism, to make up the organism. Cell is important as it performs all the functions like transportation, respiration, digestion, metabolism. All the processes which is occurring in our body is because of the cell. So that is why cell is known as structural and fundamental or functional unit of life. Let's talk about what is prokaryotic cell and what is eukaryotic cell. So when we're talking about the prokaryotic cell and the eukaryotic cell, the cell which is primitive or has been made earlier is known as the prokaryotic cell. Whereas the modifications in the prokaryotic cell or the cell which is more complex is known as eukaryotic cell. As we look at the difference between the prokaryotes and the eukaryotes, first of all, the cell that contains a clearly defined nucleus and the membrane-bound organelles. So all like we have organs in our body, cell has organelles present in the body that function like the organs in our body. So cell that has defined nucleus, that you're able to differentiate where the nucleus is, and there are organelles, they have the membrane, that is known as eukaryotic cell. Whereas in case of the prokaryotic cell, the membrane bound organelles are not present, as well as the nucleus is not clearly defined. That means there is no nuclear membrane in this. So examples of eukaryotic cell can be animal, plants, fungi, protista, whereas in case of the prokaryotic cell, it can be bacteria and archaebacteria. Size of the eukaryotic cell is bigger as compared to the prokaryotic cell. DNA replication is highly regulated in case of the eukaryotic cell as compared to the prokaryotic cell. Usually the eukaryotic cells are multicellular. That means cells can be classified into two types based upon the number of cells. One can be unicellular and the other can be multicellular. Uni means one, that means one cell will perform all the functions, whereas in case of the multicellular, there are multiple cells which will perform different function. So there are organelles present in case of the eukaryotes, whereas there are less organelles as compared to eukaryotes in case of prokaryotic cell. Let's talk about the difference between the plant cell and animal cell. So as we know, all the organisms, whether plant or whether animal, they are made up of cell. So hence the differentiation is also there of the plant cell and the animal cell. In case of plant cell, both cell wall and cell membrane are present, whereas in case of the animal cell, only the cell membrane is present, there is no cell wall. In case of plant cell, the vacuole is very big, like it constitutes 90% of the cell, which helps in the storage of the water content, whereas in case of the animal cell, the vacuoles are very small and more in number. The chloroplast is present in case of the plant cell, like plastids, they're only present in case of the plant cell, which give color. The green color pigment is present in case of the plant cell, whereas it is absent in case of animal cell. So this is the difference between plant cell and animal cell. Let's talk about the cell organelles. So there are different cell organelles present in plant cell and animal cell. So let's now talk about the cell organelles which are present. First is cell wall. So if we talk about the cell wall, so as we know, the cell wall is only one cell structure, which is not in case of the animal cell. Plants and algae have cell wall made up of cellulose. So in case of plants and algae, the cell wall is made up of cellulose. The cell walls of fungi are usually made up of chitin. So there are two cell walls. One is a primary cell wall, which is immediately outside the plasma membrane, whereas some cells produce a secondary cell wall outside the primary cell wall, known as secondary cell wall. So when a plant cell divides, there's a thin gluey layer. If you look at the diagram here, it's a thin gluey layer which is formed between the two new cells, which become the middle lamella. Next is the plasma membrane. Plasma membrane is known as a cell membrane or selectively permeable membrane. Why is it known as a selectively permeable membrane? Because it allows the selective material to pass through it and to enter. That means the selective material will be allowed to exit and to enter within the cell. That is why plasma membrane is known as selectively permeable membrane. If you look at the structure of the plasma membrane, it was S.J. Singer who gave the structure in 1972. And the structure of the plasma membrane is known as fluid mosaic model. So the eukaryotic plasma membrane has a phospholipid bilayer. So as you can see, there's a carbohydrate chain, there's a protein present here, cholesterol present here. This is the inner membrane and this is the outer membrane. So the phospholipid bilayer with proteins which are dispersed throughout the layer. So phospholipid is amphipathic, meaning it has both hydrophobic side and the hydrophilic region. There are integral proteins that have non-polar regions that completely spans the hydrophobic interior of the membrane. There are cholesterol molecules which are embedded in the interior of the bilayer to stabilize the membrane. The external surface of the plasma membrane has a carbohydrate attached to it, forming a glycocalyx. So is this a carbohydrate chain is attached? This is known as glycocalyx. So this is important for cell-to-cell recognition. So the proteins which are present in case of the plasma membrane, they provide a wide range of function. First, they transport the molecules, electrons and ions through the channel, pump and carriers in electron transport chain. Second, they act as enzymes. So there is one membrane-bound enzymes within the cell membrane that synthesize the cyclic AMP from the ATP is that adenylate cyclase. So that enzyme is known as adenylate cyclase. So there are desmosomes present which act as anchors for filament and rivet cells together. Next is nucleus. So nucleus is very, very important in case of the cell. Without the nucleus, there is half of the function of the cell is dead. Without the nucleus, there will be no function in this case. So nucleus, it contains chromosomes. So as you can see here, chromatin, thread-like structure. It contains chromosomes which are wrapped with the special protein into chromatin network. So it is surrounded by the selectively permeable nuclear membrane or the envelope that contains pores to allow the transport of molecules like there's a messenger RNA which are too large to diffuse directly through the envelope. Second is nucleolus. The nucleus of the cell in interphase consists of a region. So there's a region, this region is known as nucleolus. Here the ribosomes are synthesized. So ribosomes are synthesized in nucleolus. So one or two nucleoli may be visible. Nucleoli, they are the membrane bound structures, but actually they are tangle of chromatin. If you look at the structure here, it looks like a tangle of chromatin and unfinished ribosome precursors. So we can see in nucleolus, ribosomes are synthesized. Let's talk about ribosomes. If we talk about the nucleolus forms of ribosomes, what is ribosome? Ribosome, they are the site of protein synthesis. So ribosome, they have two units, one is smaller unit and one is a bigger unit. Smaller unit 70S and the bigger one 80S. So ribosomes synthesize proteins. There is another place from which the ribosomes are being produced, the proteins are being synthesized, that is known as endoplasmic reticulum. So let's talk about endoplasmic reticulum. Endoplasmic reticulum is a membranous system of channels and flattened sac that traverse the cytoplasm. So all these organelles, if you have the structure of the cell, this is the nucleus. Outside the nucleus, cytoplasm is present. In the cytoplasm, all the organelles are there present. So cytoplasm is jelly-like substance, so the liquid where all the cell organelles are present. So if we talk about endoplasmic reticulum, there are two types of endoplasmic reticulum. One is smooth endoplasmic reticulum and the second one is rough endoplasmic reticulum. Rough endoplasmic reticulum, why is it rough? Because there are some rough substances present over the surface. That rough substances are known as ribosomes. Whereas if we look at the smooth endoplasmic reticulum, the surface is smooth because there are no ribosomes present on it. So as we know, the function of the ribosome is to synthesize proteins. That means rough endoplasmic reticulum synthesize proteins. Whereas in case of smooth endoplasmic reticulum, it has three functions. One, it synthesizes the steroid hormones and other lipids. Second, it connects rough endoplasmic reticulum. So this rough endoplasmic reticulum synthesizes proteins and which is attached to the ribosomes. Second is smooth endoplasmic reticulum. Smooth endoplasmic reticulum assists in the synthesis of hormones and lipids. So like the rough endoplasmic reticulum synthesize proteins, smooth endoplasmic reticulum synthesize lipids. Third, it connects rough endoplasmic reticulum to the Golgi apparatus. So the smooth endoplasmic reticulum is connecting the rough endoplasmic reticulum to the Golgi apparatus. That is one of the other organelle which we will talk about. Third, it carries out the various detoxification processes. So this is the function of rough and the smooth endoplasmic reticulum. Next is Golgi apparatus. So as we have just studied that smooth endoplasmic reticulum joins rough endoplasmic reticulum to the Golgi apparatus. So Golgi apparatus was discovered by the scientist Camillo Golgi. So it is a membranous, what we can say is, it lies near the nucleus and consists of flattened membranous sacs stacked next to one another, and it is surrounded by the vesicles. So as you can see, these are the new vesicles which are being formed, right? So what is the function of Golgi apparatus? That package the whatever protein is getting and it is acting like a courier system. So it is packaging the substances produced in the rough endoplasmic reticulum and secrete them to other cell parts of the cell surface for the export. Next is lysosomes. So lysosomes, they are known as suicidal bags. So let's study why lysosomes are known as suicidal bags. They contain hydrolytic enzymes, or they are known as digestive enzymes, which are surrounded by a single membrane. So what they do is they have those hydrolytic enzymes to kill the foreign material, the waste material present in it. So why it is known as suicidal bags? Because if the cell gets damaged, the content will burst out, and hence the hydrolytic enzymes will also come out, and those enzymes will kill their own cell. That is why it is known as suicidal bags. So they are the principal side of intracellular digestion. So digestion takes place in lysosomes. So with the help of the lysosome, the cell continuously renew itself by breaking down and recycling cell parts. That is with the help of the lysosomes. Next is mitochondria. So as we know, mitochondria is known as powerhouse of the cell. It is the site of cellular respiration. It is also known as powerhouse of the cell. That means it has the most important function. Let's study about it. So mitochondria, the site of cellular respiration. All cells have many mitochondria. They are active cells. It could be many of them. So mitochondria have a double, there's the outer membrane and the inner membrane. Inner membrane, as you can see, it's highly folded inside. It has a matrix present in it. So in case of mitochondria, they have their own DNA, and they're known as powerhouse because it produces energy in the form of ATP, that is adenosine triphosphate. Next, we have the vacuole and plastids. So as we have discussed when we were differentiating the plant cell and the animal cell, the vacuole is bigger. It contains 90% of the cell part in case of plant cell, whereas in case of the animal cell, they may be present like very small and it is very less. So vacuoles are single membrane bound structures for storage. There are vesicles present inside the vacuoles. Protista, exception is there. Protista, they have contractile vacuoles. So with the help of contractile vacuoles, they can pump out the excess of water. Next is plastids. So when we were discussing the differentiation, as we know that the green color pigment chloroplast is a type of plastid. So plastids are only present in case of the plant cell. It is not present in case of the animal cell. It is divided into chromoplast, chloroplast and leucoplast. Chromoplast, which gives the color. So like a chromoplast, they store the carotenoid pigment. They're responsible for red, orange, yellow color of carrot, tomatoes and daffodils. Whereas in case of the chloroplast, they're the site of protein synthesis. In addition to the double outer membrane, they have inner one also. As you can see, the outer membrane, inner membrane, stack of stroma, thylakoids, lumen and granum. So grana lies in the stroma. They contain their own DNA. And the third one is leucoplast, which stored starch and are found in roots like turnips or in tubers like potato. So this is about the cell organelles. Let's try out some important questions that come in exam from the cell unit. Okay, so all of these questions are very important as we have discussed in the cell part. First, which organelle produces ATP? So if we see out of all of these, it's mitochondria. So we can say mitochondria produces ATP because it is known as powerhouse of the cell. Which produces protein? As we know, the proteins are produced by ribosomes, and ribosomes are attached to rough endoplasmic reticulum. Third, which package and secretes the substances? That's Golgi Apparatus. So all this product of endoplasmic reticulum, rough endoplasmic reticulum, is packaged and secreted by Golgi Apparatus. Next, which contains hydrolytic enzymes. So hydrolytic enzymes are used to kill the foreign material or the waste material. That is by lysosomes. Which directly assist with cell division, that is microtubules. There's a 9+2 arrangement, 9+1 arrangement of this. So microtubules, they help in directly assisting with cell division. Next, look at the question. Which of the following is not normally found in a plant cell? So as we have talked about the difference between the plant cell and the animal cell, which is not present in case of the plant cell, that is mitochondria is present, endoplasmic reticulum present, plastids only in plant cell, plasma membrane is also present, centrioles. So centriole is not present in case of plant cell. Instead they have the microtubules. So instead of centrioles, plant cell have microtubules. Ribosomes, they have all other mitochondria, ribosomes, plastids and endoplasmic reticulum. Next, which of the following is present in a prokaryotic cell? So which is present in prokaryotic cell? It's ribosome. So as we know, the complete organelles are not present in case of the plant cell, like mitochondria, endoplasmic reticulum, chloroplast, nuclear membrane, all of them are absent. Ribosome that synthesize the proteins are present in case of prokaryotic cell. Next, smooth endoplasmic reticulum. What is the function of smooth endoplasmic reticulum? As we have discussed, it detoxifies the alcohol in the liver cell. Then centrioles. So as we have just discussed, centrioles, what they do? Consist of microtubules. Peroxisomes, they break down the product of cell respiration. nucleolus. As we can see, nucleolus and ribosomes. Lysosomes, as we know, they digest the damaged cell. So smooth endoplasmic reticulum, what they do, they detoxifies the alcohol in the liver cell. Whereas, if we talk about the centrioles, it consists of a microtubule, that is with a 9-triplet configuration. Peroxisomes, they break down the product, that is peroxides, and centrioles is breaking down the product, peroxisomes. Then nucleolus is for synthesizing the ribosomal RNA, whereas the lysosome, they digest the damaged cell. So this is about the cell and the different organelles present. I hope you understood. Thank you.